Hi, welcome everyone to this month's episode of our monthly seminar series at the Society for Functional Precision Medicine. I'm Tony Latai, president of the society. Before we get started, I just want to remind everyone of a couple things. One is you are on this recording and you are not a member, please join. I will remind all trainees that joining is free for you, but it is nonetheless very important for us. We want to know who you are and we want to build our membership. So join yourselves and bring a friend along. Also, a reminder that in September, we have a very exciting meeting in Copenhagen, September 25th in Copenhagen. You can read about it on our website, sfpm.io. It's joint with EHA, the European Hematology Association. I think it's to be a great meeting. Actually sort of historic. It's the first meeting of its kind, joining the functional and the genomic precision medicine fields in a single meeting. So I'd encourage you all to check that out and attend the meeting if you can. So for today, before we get to our main speaker, Constantine Mitsiades, Tanya Bondar from Blood Cancer Discovery has been kind enough to join us just to tell us a little about her journal and to try to foster a relationship between her journal and the members of SFPM. I've published in Blood Cancer Discovery. It was a very good experience, and I think you guys should consider it, especially for your blood cancer results. So without further ado, I'll let Tanya take it away, and then I'll introduce Constantine after she's done.
Thank you so much, Tony, for the introduction and for the opportunity. Blood Cancer Discovery is one of the AACR journals, and we're really interested in functional precision medicine. The journal is relatively new. It's been launched in 2020, and last year we got our first impact factor of 11.4, which is comparable to what our sister journal Cancer Discovery had as the first impact factor. So we're generally following their successful model. The choice of where to publish your work is more complicated than just going by the impact factor. There is this other big part of the decision, which is not as easy to measure by any metrics, and it is author service. So I will break it down a little bit more in the next slide. But here I'll just want to mention that the model of cancer discovery and blood cancer discovery is grounded on the synergy between academic editorial board, stellar experts in their fields, our editors-in-chief, and an in-house team who has more time to make sure things are moving timely. So that together supports the author service. So the things to consider, and that we try to be above competitor journals in providing good author service, is ease of initial submission. You can just send the pre-submission inquiry first. We don't require any specific formatting in the first consideration. We try to be as fast as possible with our decision time. So from submission to acceptance and online publication of your paper, the average time is under 6 months, and that includes 3 months that we give for a revision. We make decision based on expertise of our academic board. And you can look up on the journal website that they're really amazing experts in their fields. We make sure that peer review is constructive, and that we try to find a home for every paper that goes out for review, either in our journal or in a sister journal. There is an easy transfer system between manuscripts within AACR family, the post-publication promotion of our content is something also that we're quite proud of. So these are different things we do. Some of them we do for every paper. Like every paper gets opener art. It's like a journal cover designed uniquely for your story, and we tweet about it. But then there are also ways to promote publications through article collections, ACR blog, concurrent publications with major conferences. We commission commentaries for about 50% of articles. And these are just a few examples of what we've published in the functional precision medicine field. So I encourage you to not rely on just my words, but if you know these people, which I'm sure some of them you do, you can ask for their experience with the journal. That's pretty much all. Oh, and just following what Dr. Litai said about the functional precision medicine meeting in Copenhagen. So if you are planning to submit an abstract to that meeting and you would like to have your work published at the same time with your presentation, please consider sending it to Blood Cancer Discovery by the end of the month. Of course, if it is about blood cancers. And we cannot promise that it would be accepted. We're quite selective, but we will make sure that the editorial decision will be timed with the conference. So articles that are published together with presentation at conferences, they generally get more attention. And so on this slide are just some contact links and emails, and I also put it in the chat. And please feel free to reach out to me about any questions or discuss any potential submissions. Thank you very much for your time.
Oh, thank you very much, Tanya. As I say, I found the journal very easy to work with, and you might too, so you might consider it. Now I'd like to, Constantine, you can start sharing your slides, I think. I'd like to introduce my colleague, Constantine Mitsiades, who's an associate professor in the Department of Medical Oncology at Dana-Farber and also an associate member of the Broad Institute. He got his MD at the University of Athens Medical School in Greece. He focuses on understanding mechanisms of resistance to both drug as well as immune therapies, with a focus in myeloma, but also other cancers. He is particularly interested in non-genomic mechanisms of treatment resistance, which I think is very important, and I suspect is probably the most important aspect of drug resistance in cancer. And he's observed this both as a result of drug treatment as well as a result of immune treatment. Today, he's going to be talking about some CRISPR screens he's applied to dissect out essentiality in certain tumor types that are harder to identify by standard omics approaches. You can put your questions, as always, in the chat or ask them in person. So, Constantine, take it away. Hey, Aaron, I see some black bars on the I see some black bars on the screen. Do you know what that's all about? Are you seeing that, Constantine?
I am seeing right now that the meeting controls have been deactivated. So I think now you shouldn't be seeing them.
Okay, I still see some black bars, but it's not too bad. But go ahead. I don't want to interrupt further.
Yeah, so Tony, thank you for the kind introduction, and it's a pleasure for me to be part of the Society for Functional Precision Medicine and be able to present on this monthly series. What I was hoping to do today is give to you an overview of our recent experience on using CRISPR-based functional genomic studies to identify lineage-related dependencies that can play preferential roles in individual types of malignancies. Most of what I will describe as data come from a recent publication that we had that was focusing on multiple myeloma, but I will also describe some data that were part of that effort that were relevant to other neoplasias, and I hope that what you will hear will be a great food for thought and an opportunity for collaborations within the SFPM and beyond. So the first slide is probably the least important, shows my commercial disclosures, none of which have, to my understanding, an impact on the content of this presentation. The next slide is probably the most important. It is to acknowledge the different members of my lab and collaborators that contributed to this work conceptually and experimentally and analytically. I do have to, of course, acknowledge the colleagues who are listed with asterisks: Ricardo D'Amato Simoes, Ryosuke Shirasaki, Jeff Matthews, Sondra Downey, and Ben Barwick. These are the co-first authors of a manuscript that we had last year on applying the concept of preferential dependencies in multiple myeloma. These were members of my lab, and Ben at the time was a member of the lab of Larry Boyes at Emory. And this was a great collaboration with the Dependency Map program at the Broad, Paquita Vasquez, Aviet Cherniak, and other members of the Broad. And this was also a setting and a project for which I do need to give a lot of thanks to Larry, because Larry Boyes was not only a collaborator for this particular project and a key member of this multi-institutional team, but also because, as you will hear during the presentation, earlier work from his studies was a great influence and a great stimulus for this particular project. And I also want to thank Tony, not only because he is leading the SFPM, but because in many ways what you will hear in this presentation has been influenced by the discussion that Tony and I have had over the years about how we should not just focus on standard omics to characterize candidate dependencies, but look into function as our guiding principle on identifying new therapeutic targets and promising therapeutic targets. So my laboratory focuses on treatment resistance. As Tony mentioned, this is an overview slide that gives you a snapshot of what types of systems we've been using to address genomic resistance with CRISPR and PRISM screens, how we have been trying to assess non-genomic persistence. In the last few years, we have had the good fortune to report in our studies on how minimal residual disease after cytotoxic chemotherapy in solid tumors or heme malignancies can be associated with a transient non-genomic persistence state that resembles embryonic diapause. And as some of you know, and we've had the good fortune also in collaborating for some of these studies with Tony, we've been very interested in microenvironment-mediated resistance as an additional non-genomic mechanism of resistance. We have reported in multiple myeloma, but other neoplasias, that when tumor cells interact with non-malignant accessory cells, for instance, bone marrow mesenchymal stromal cells, this interaction can protect the tumor cells from various pharmacological or immune-based insults. And in fact, in one of the studies that we had the chance to collaborate with Tony, this interaction was documented to be associated with a change in the BH3 profiling of the tumor cells, and specifically a decrease in apoptotic priming, which can explain in many ways why these interactions with accessory cells can have such a profound impact in attenuating treatment response in many different settings. We have tried to incorporate all of these different platforms for in vitro and in vivo assessment of genomic and non-genomic mechanisms of resistance, and we have had the good fortune to see some of this work allow us to define rational combination regimens, rationally designed combination regimens that have made it into the clinic successfully. And some of these therapies have been FDA approved or became standards of care for multiple myeloma. We hope to be building more on this work with some of our new systems in the years to come. As Tony mentioned, we've been interested in how genomic and non-genomic mechanisms of resistance and persistence apply to NK cell therapies, and we've published some of this work recently. We've been interested in the biology of minimal residual disease, and what I will describe to you today is the work that we published last year in terms of preferential dependencies from multiple myeloma. We became interested in this idea about a decade ago, thanks to a review article that was written by Larry Boyes and was published in Blood. At the time that Larry wrote this review, there have been approximately 15 years of very rapid clinical development in myeloma of a series of therapies that have truly revolutionized the therapeutic outcomes in this disease. What Larry pointed in his review was that all of these therapeutics that have shown major activity in myeloma and were developed between the end of the 90s and 2014 did not really target mutationally activated processes in myeloma cells. Instead, they targeted biological processes and specific targets that are highly important for myeloma, highly important for plasma cells, but don't necessarily distinguish a malignant from a normal plasma cell. For instance, IKZF1 and IKZF3 are targeted by thalidomide derivatives. Proteasome inhibitors target NF-kappa B endoplasmic reticulum stress. CD38 and SLAMF7 are targeted by monoclonal antibodies that are FDA approved. And all of these pathways and respective targets are important for myeloma cells, but they are apparently also important for normal plasma cells. And an important aspect that was highlighted in the review was that these processes were not only highly important for myeloma and malignant plasma cells and maybe normal plasma cells, but also that these therapies had limited clinical applications beyond plasma cell neoplasias, presumably reflecting that the role of these respective targets was limited, if any, in these other lineages. And in the years that followed, in this decade that followed, several additional therapies for multiple myeloma were developed. For instance, CAR-Ts or bispecific T-cell engagers against BCMA or GPRC5D. And again, these are plasma cell-specific antigens conforming to what Larry described in his review about a decade ago. I'm also including on this slide BCL2 and HDAC1 or 3. Inhibitors for these targets are not currently FDA approved for multiple myeloma. This is probably due to unfortunate circumstances related to the clinical development of these agents. There were unfortunately no randomized clinical trials documenting benefit for BCL2 inhibitors in myeloma. For the HDAC inhibitors, there were 2 such trials, but with rather modest clinical benefit and a substantial profile of adverse events. But I am including these particular targets on the slide because despite the lack of FDA approval or randomized clinical trial data, these agents are more active in multiple myeloma than the overwhelming majority of other neoplasias. Clearly, a lot more activity for BCL2 inhibitors in AML, CLL, and other lymphoid malignancies. But also clearly the activity that we see and have seen in multiple myeloma with HDAC inhibitors was far more pronounced than any other setting where these agents were tested. So you can clearly make the case that the clinical activity of HDAC inhibitors and to some extent BCL2 inhibitors is still preferentially important for multiple myeloma compared to other neoplasias. I also will give you a preview by saying that BCL2 and HDAC1 are 2 genes that were identified in our CRISPR studies as preferential dependencies for multiple myeloma, and I will get back to that point later in the presentation. So this review, this highly influential review, led my lab and I to ask the question, if so many or nearly all of the highly successful therapies recently developed in myeloma target both malignant and normal plasma cells alike, can we identify other dependencies that may not distinguish between malignant and normal plasma cells, but can be very important for myeloma therapeutically because the adult patient can survive if these molecules and processes are targeted, because the adult patient can survive without active normal plasma cells for extended periods of time. An indirect piece of evidence that supported the validity of this statement comes from the fact that some of the therapies that we just heard about actually have activity not only in plasma cell malignancies, but in non-oncologic conditions in which there are non-malignant but abnormal pathogenic plasma cells. And an example of that is the case of proteasome inhibitors, which have been shown over the last decade to have activity, clinical activity in different settings of autoimmune conditions, for instance, systemic lupus erythematosus. And this clinical activity correlates with the extent of suppression of the non-malignant but pathogenic plasma cells of this condition. And so this was an additional reason for us to feel confident that if we identify therapies that are preferentially essential for plasma cell malignancies, these are probably going to be the types of therapies that will be tolerated, or at least it will be feasible to administer them to patients without catastrophic toxicities. And so another consideration that I think is very important for everyone on this seminar, even those who have no direct interest in multiple myeloma, is that if we really want to consider what have been some of the most successful therapies we've used across any type of cancer, these actually happen to be lineage-specific therapies. Anti-estrogens and other hormonal manipulations for ER-positive breast cancer, androgen ablation therapy for prostate cancer, radioactive iodine therapy for thyroid cancer, even myeloablative hematopoietic stem cell transplantation for hem neoplasias. These are all lineage-specific therapies. They target both malignant and normal cells of a given lineage, but they spare the overwhelming majority of other tissues, and they cause limited or no major life-threatening complication in an adult patient. In fact, those of you who may recall, many years ago, we were referring to, for instance, hematopoietic stem cell transplantation as high-dose chemotherapy or megatherapy for those of you who may have worked in Europe. But we also always attach the term hematopoietic stem cell rescue, because really the hematopoietic stem cell administration is to rescue the complications that are associated with the lineage-specific myeloablative chemotherapy that eliminates both normal and hopefully the malignant tumor cells. We feel that this is a concept that is very relevant not only for the diseases that are mentioned here on the slide and multiple myeloma, but other hematologic malignancies, other cancers of reproductive organs, other endocrine neoplasias, but potentially can be relevant for neoplasias that involve the kidney, the pancreas, or neoplasias of adipose tissue like liposarcomas, because in principle, at least, any therapy that would be able to ablate normal and malignant cells from these lineages would likely be associated with some toxicities, but probably the hope is that if conducted appropriately, the clinical administration of those agents can be associated with no major life-threatening complication. Of course, this idea of lineage-specific therapies is not readily applicable in situations where normal counterparts of a given tumor cannot be safely ablated, or in those situations where selective therapies against lineage-specific dependencies are lacking. And so this is an important consideration to keep in mind, and I think we will have hopefully opportunity to further discuss this today during the Q&A session. So building on this wonderful and very insightful observation from Larry and his colleagues, we decided to make a systematic assessment of the dependencies of myeloma cells in collaboration with the DEPMAP program at the Broad. And the goal was to try to identify through genome scale, CRISPR-based knockout screens, what are the patterns of dependencies in multiple myeloma cells, what are the patterns of dependencies in other lineages or tumors of other lineages, and identify those genes that were following a pattern similar to the red cells that you see on this diagram. The knockout that corresponds to this red cell is depleted from the cultures of multiple myeloma cells, but does not have any detrimental impact on the in vitro fitness of tumor cells from other lineages in this schematic representation. This is the type of preferential dependency that we are looking for, preferentially essential genes for multiple myeloma cells. Perhaps these are the types of genes that are equally important for normal plasma cells, but for the reasons outlined earlier, this is not something that creates necessarily a prohibitive issue for further consideration. And I want to distinguish the knockout that is represented here in the red cells from the knockouts that are represented with the green cells that are detected at the start of the treatment, or at the start, I should say, of the genome scale CRISPR screens for all of the tumor cells of these different lineages. But you will see that the green cells are completely depleted, not only on the myeloma, but also on the other blood cancer and solid tumor screens. These are the types of genes that may perhaps be also reasonable therapeutic targets under certain conditions, but these are the types of genes that we would consider as core essential or broadly essential genes. And in order to feel comfortable that they are tractable therapeutic targets, they would have to be the types of genes where pharmacological inhibition can be achieved without catastrophic toxicities to normal tissues. So The results that were generated by this particular effort can be summarized in this one slide, where you see these two heat maps. And they present the results of the essentiality scores that were identified for these 116 genes that met all the selection criteria that are outlined on the left-hand side of the screen. So these essentiality scores are the so-called series scores. They're quantitative metrics of relative dependence of a given human tumor cell line on a particular gene in the absence of a drug treatment. You will notice that the black and dark blue colors represent negative series scores. This means pronounced depletion of the guide RNAs for the knockout of a given gene in a particular cell line. That means more pronounced essentiality. And on the right-hand side of this double heat map, you see multiple myeloma cell lines and the data for their essentiality scores for these 116 different genes depicted as a matrix, where each cell line is represented in a different column and each gene is represented in a different row. On the right-hand side, you have this second heat map in which the essentiality scores for each gene are presented in a row in a stacked bar graph format. And the reason that this is done in this fashion is because we wanted to visualize the essentiality scores for each gene for the non-myeloma lines from the lowest essentiality on the left to the highest essentiality on the right. And we did this in order to provide an opportunity for these dual heat maps to give us direct visual information whether a gene is highly essential for the multiple myeloma cell lines on the right, with lots of black and dark blue color, and conversely, be in position to easily see that there is absence of color for the majority of the non-myeloma cell lines on the left. And lack of color here means lack of essentiality. I mentioned a moment ago that we identified these 116 genes based on a series of selection approaches. The criteria outlined in more detail on the paper, but basically we took into account the genes that had a significant magnitude of differential dependency between myeloma and non-myeloma, both in terms of the difference in these series scores for essentiality, as well as Fisher tests that tried to allow us to filter out those genes that may have statistically significant difference in the essentiality scores, but the effects were quite small. We looked into genes that had to meet some standard criteria for the strength of the dependency within the multiple myeloma cell lines. We used additional criteria to filter out those core essential, globally essential genes. And these genes obviously had to have some recurrent expression in multiple myeloma cell lines in order to be considered. The data that we presented in the paper were the product of analysis that took into account several different depth map releases, and we observed a very nice concordance of results across several of the later release of the depth map data. And also for those of you who may follow the literature and developments in the area of genome scale CRISPR screens, we did follow the series scores. We have since confirmed that the overwhelming majority of these 116 genes meet all the criteria for preferential dependencies if you use additional essentiality scores such as the Chronos scores. And also we took into account data that do not include the copy number correction that's inherent to the series or the Chronos scores in order to make sure that the identification of the data for these preferential dependencies was not in any way confounded by the specific type of DNA copy correction method that we applied. So this slide was showing to you these 116 myeloma preferential dependencies in a way that involved a ranking from top to bottom in terms of the relative essentiality, the differential essentiality in multiple myeloma versus other cancers. On this slide, in order to make it also a little bit easier visually for us to see the gene symbols, we tried to cluster these preferential dependencies in terms of the functional groups that they are part of. And you will notice on the left hand side of the slide that we have this same dual heat map presentation with myeloma lines as a matrix on the right and non-myeloma cell line data on the left. You will see in this large red box a major collection of transcription factors, cofactors, transcriptional regulators, RNA binding proteins, splicing regulators, chromatin remodeling genes. This is the most profound group of genes that are identified as myeloma preferential dependencies. And as you may see on our manuscript, on those instances that we described preferential dependencies for other cancers, it is generally an observation that the preferential dependencies for different types of cancers tend to be highly enriched for transcriptional regulators and especially lineage-defining transcription factors. So this is something that's very consistent across many types of cancers. And of course, the individual partners, the individual transcription factors, cofactors, and chromatin remodeling genes are distinct, with some cases of overlap between highly similar or cancer types that share a lot of biological similarities. Very distinctly for myeloma, we had this large cluster of genes that are involved in unfolded protein response, endoplasmic reticulum stress, and Golgi function. This is very consistent with the knowledge we have in the field of multiple myeloma, that this is a disease that is exquisitely dependent on endoplasmic reticulum stress because of the need of these cells to be able to properly fold the large amounts of immunoglobulin protein particles that they are synthesizing. And because this process is not perfect, there is a lot of misfolded immunoglobulin proteins that are being synthesized, and that makes myeloma cells highly responsive and highly susceptible to any disruption of their endoplasmic reticulum stress. There are also other groups of genes that I would like to highlight, very relevant to our conversation today, and because it does happen that the BCL2 family is very near and dear to the hearts of many of us on this forum, including both Tony and Larry. Mitochondrial regulation of apoptosis was represented in this list of preferential dependencies with MARCH-F5 and also BCL2. As I had mentioned earlier in this presentation, although unfortunately BCL2 inhibitors have not yet reached what we all had hoped for, their FDA approval for multiple myeloma, there's clear evidence that there are subsets of myeloma patients with great clinical response to BCL2 inhibition. And I in many ways consistent with that, BCL2 as a gene in vitro in myeloma cell lines is a preferential dependency compared to the overwhelming majority of non-myeloma tumors. And the same thing also applies for HDAC1, which was listed within the chromatin remodeling genes that I mentioned earlier. Other categories of genes that were represented here, many different types of signaling genes relevant to NF-kappa B, upstream regulators, insulin-like growth factor receptor signaling, IL-6 signaling, FGFR3, and other pathways, many different types of metabolically relevant genes, and other categories of genes, some of which have not been extensively characterized yet in multiple myeloma or beyond, or genes that may have multiple different possible interactions with some of the functional groups that were mentioned earlier. What I would like to emphasize is the fact that while many of the genes that are present in this list have known roles in the biology of myeloma, IRF4, XPP1 as lineage-defining transcription factors, MAF and FGFR3 as genes that are recurrently dysregulated in this disease, there are many other myeloma preferential driver genes that were identified from this exercise that were previously understudied. And very importantly, essentially all the mediators and targets of drugs that have demonstrated more potent clinical activity in myeloma compared to other neoplasias were represented in this list. IKZF1, IKZF3, transcripts for transcription factor members of the NF-kappa B family, the endoplasmic reticulum genes, both NF-kappa B and endoplasmic reticulum being targets of proteasome inhibition, all of these drug targets and mediators of activity for these preferential therapeutics, preferential drugs with preferential clinical activity in myeloma were represented in this list. And this is very reassuring because it gives us more confidence that some of the previously understudied genes identified from this effort may also represent promising therapeutic targets. Finally, on this slide, I want to mention that one of the chromatin remodeling genes, EP300, and one of the signaling genes, PIM2, are very intriguing because there have been clinical trials of pharmacological agents that have demonstrated promising results in this disease. Unfortunately, for the PIM inhibitors, these were non-specific PIM inhibitors. They were also targeting PIM1 and PIM3, not only PIM2. In fact, they were not designed to hit PIM2 with great potency, but still they demonstrated promising clinical activity in relapsed refractory myeloma during this last decade. And we hope that based on the data from this paper and other studies, there will be a renewed interest in developing PIM2-specific inhibitors for multiple myeloma. P300, the protein encoded by EP300, is a target for inhibitors that were recently shown to have promising clinical activity as single agent, but especially in combination with thalidomide derivatives. These results were presented during this last year, almost concurrently with the results from these CRISPR screens, and we're very excited about this observation because, again, this is another indication that some of the genes that are not yet targeted with FDA-approved agents, but are represented in this list may actually have great potential for therapeutic applications in myeloma. In these studies that we conducted, we also tried to perform additional functional validation studies. For instance, we validated the extent to which these myeloma preferential dependencies are also required for the survival and growth of human myeloma cells in vivo. We examined this question in a specialized ceramic scaffold system in which we take these bicalcium phosphate scaffolds and culture them ex vivo in the presence of human mesenchymal bone marrow stromal cells from donors or patients, and under conditions that favor the osteogenic differentiation of the multipotent cells that are present in this population. And what that does is that it promotes the osteogenic differentiation of these multipotent cells so that when these scaffolds are implanted subcutaneously in mice, the osteogenic cells are producing a bone-like material that coats the surface of the scaffold and creates a sinusoidal structure that resembles the human bone marrow sinusoids. And importantly, because the stromal compartment and osteogenic cells in that system are human, the cytokines that they produce are providing a better support system for human cell lines and patient-derived xenografts. And in fact, that's how we have used this system. That's why the system was originally developed by my former postdoc, Richard Kroon. In this case, we didn't use it for expansion of PDX cells, but we use it in order to test two different multiple myeloma cell lines, KMS11 and XG7, which were engineered to stably express the Cas9 nuclease, were transduced with a focused library of guide RNAs against a collection of genes, including 88 of these myeloma preferential dependencies. The tumor cell populations were injected directly into these scaffolds, and then next generation sequencing allowed us to confirm that the knockouts for these genes were significantly depleted. Basically, pretty much all of the genes that were myeloma preferential dependencies that were included in this focused library were essential for at least one and typically both of the cell lines that were examined in this particular in vivo experiment. One of the advantages also of this exercise was that while it validated certain pieces of knowledge that we kind of expected, the notion that IKZF1 and IKZF3 were going to be essential for multiple myeloma cells, it also gave us new clues about some of these areas of previously held knowledge. For instance, thalidomide derivatives don't just interact with IKZF1 and 3, they also interact with many other substrates. And the question has always existed over the last decade. Okay, IKZF1 and 3 are great targets for thalidomide derivatives, but what's the role of the other substrates or targets? Could it be that multiple myeloma cells respond so well to these drugs that exclusively because of the IKZF1 and 3 effect, or do some of the other therapeutics, some of the other potential therapeutic targets play a role? And because thalidomide derivatives have limited applications beyond plasma cell dyscrasias, we reasoned that the genes, the targets that are the most likely mediators of the effect and the preferential clinical activity of this drug class in myeloma have to also be preferential dependencies. And indeed, IKZF1 and 3 are at the top of this list of the different known or reported substrates for and interactors for thalidomide derivatives. What was interesting, though, was that we identified another molecule called ARID2, which was reported over the last 3 to 4 years to be a substrate for thalidomide derivatives like pomalidomide, but not lenalidomide or thalidomide, the earlier versions of this drug class. And so we reason that this is a very interesting observation here, because it may explain why pomalidomide tends to be more potent than lenalidomide and thalidomide. And also it highlights the significance of directing the efforts for creation of new thalidomide derivatives towards not only IKZF1 and IKZF3, but perhaps ARID2 in order to be able to better simulate this effect and leverage the preferential essentiality of ARID2. These results do not mean that the other known or proposed targets and mediators of effects of thalidomide derivatives are not important, but they just mean that they cannot explain on their own the preferential nature of the clinical activity of thalidomide derivatives in myeloma. In the paper, we also examined several other genes and did extensive functional analyses for their role in multiple myeloma. I encourage you to take a closer look into the studies about POU2F1. In that paper, POU2F1 is a key transcriptional cofactor for myeloma cells and the top preferential dependency among the transcriptional cofactors. We validated with CRISPR interference and CRISPR activation methods that this is a gene that's important for multiple myeloma cells. So the CRISPR editing results of the primary screen were further validated with 2 additional functional genomics methods. And again, in collaboration with Larry and Ben Barwick, we documented that there is an enrichment of POU2F1 binding motifs in loci proximal to myeloma preferential dependencies, suggesting that part of the mechanism of how POU2F1 serves as a preferential dependency is by regulating the expression of other multiple myeloma preferential dependencies. And we also conducted extensive studies on different types of endoplasmic reticulum related myeloma preferential dependencies. This is an example of UBE2J1, which is an E2 conjugating enzyme that's resident to the ER. And there are several other genes that we identified in this study, and they're highlighted here on the schematic diagram in blue, including synoviolin, HERPUD1, CEL1L, and different glycosylation regulators that are part of the glycoprotein quality control system of the ER. Perhaps the most important message that we derived out of this work is that when we look into this collection of myeloma preferential dependencies, they are not among the top genes that are recurrently mutated, recurrently amplified, or transcriptionally dysregulated in multiple myeloma. For example, if you look at this dual heat map, for the top 200 genes with overexpression in multiple myeloma versus non-myeloma cell lines, you will notice that, yes, there's a collection of myeloma preferential dependencies that are highlighted with their respective symbol, and it's 17 genes within this top 200 most overexpressed genes in multiple myeloma. What this means, though, is that the large majority of the top overexpressed genes in multiple myeloma are not preferential dependencies. And in fact, unlike the heat maps that you saw earlier, there is very limited blue and black in this myeloma part of the heat map. Most of the top overexpressed genes in myeloma versus non-myeloma cells are not dependencies at all in this disease. So this is very important to keep in mind. And when we look into other omics data, like copy number gain data in multiple myeloma patient samples, or the frequency of mutations in different genes in multiple myeloma patients, you will notice that there's 4 or 5 genes that are amplified in the top 200 most amplified genes. And there's only a couple of multiple myeloma preferential dependencies that are within the top 200 or so most frequently mutated genes in this disease. When you look into correlation with clinical outcome, this is a complex matrix that tries to summarize the results of overall survival or progression-free survival in 6 different studies for these 116 preferential dependencies. And basically, wherever you see white, it means that that particular gene in that particular study did not meet criteria for statistically significant correlation with outcome. Wherever you see red, there is a significant correlation with adverse outcome. Wherever you see blue, significant correlation with favorable outcome. So you will notice that some genes have some red boxes, a few genes have some blue boxes, but these results are not highly consistent. For some genes, they apply only for progression-free survival, only overall survival, or only some of the studies. And when the results look like they're concordant between overall and progression-free survival, these are typically the types of genes that are overexpressed in the context of some other genomic event that correlates itself with adverse outcome. And so it's not totally clear that this particular gene is a driver of adverse outcome, but it is a preferential dependency. And the bigger theme here to remember is that if we aggregate all of these different omics data and try to present them in this circus plot, where the center of the plot highlights the frequency of mutations and copy number gains, the transcriptional upregulation of these 116 preferential dependencies in myeloma versus non-myeloma lines or patient samples, the overexpression in myeloma versus normal plasma cells, the overexpression in late-stage disease, such as plasma cell leukemia versus early-stage myeloma, or when we compare patients with shorter progression-free survival. All of these types of omics-related metrics that we've been using to identify promising candidate dependencies in different cancers, they seem to be relatively quiet. And this is to us, and there are, of course, some exceptions to that, but overall, these are the types of genes that we have every reason to believe that they are very relevant for cell lines, they're very relevant for early stage and late stage disease, for high risk and standard risk myeloma, because they target the plasma cell-related biology of the disease, and they're less related to the mechanisms that lead to the establishment of the disease in the first place. And if you go back to Larry's tau of myeloma review, in this yin and yang relationship, if the plasma cell-related biology of myeloma is what is represented by these preferential dependencies, the cancer-related biology of myeloma is the key determinant of high risk in this disease. And of course, ultimately, we may want to target both of these aspects. But right now in multiple myeloma, we're doing a much better job of targeting the plasma cell-related biology. And based on these 116 preferential dependencies, we have reason to think that we have a plethora of promising targets to continue targeting this aspect of the biology very efficiently until we can get better at targeting the high-risk aspect of the genomic feature of the disease in late-stage patients. Going back to this slide that I showed earlier about lineage-specific therapies, I wanted to highlight that we did a similar analysis for preferential dependencies in other neoplasias following the same set of criteria that we applied in multiple myeloma. And what you will notice here is that if you rank on this dot plot the number of preferential dependencies identified for each type and plot that versus the number of lines for each tumor type that was evaluated, you will notice that myeloma has a very high number of preferential dependencies. There are other diseases like AML or lymphomas that also have a high number of preferential dependencies, but there doesn't seem to be a great correlation between the number of preferential dependencies that are being identified and the number of cell lines that you need in order to get there. Of course, the more cell lines you examine, the higher the number of preferential dependencies that you will identify. But we think that what this slide here highlights is this notion that Maybe preferential dependencies are not as easy to identify for certain types of cancers, especially the ones where ablation of their respective normal cells is going to be more difficult to perform. And also another consideration here to keep in mind is that multiple myeloma cells have a very distinct set of biological pathways and transcriptional networks that they need to apply in order to execute their functions. They have a very distinct transcriptional network compared to other heme malignancies, which also have a very distinct set of transcriptional networks compared to other neoplasias. And so we feel that it stands to reason why multiple myeloma and to some extent other heme malignancies can allow us such a straightforward, relatively easy identification of preferential dependencies, and that much larger series of cell lines may be needed for some other types of tumors. For those of you who have an interest in some of these other neoplasias, these are examples of dual heat maps that are included in the supplement of our paper in terms of AML preferential dependencies, genes that are preferentially essential for ER-positive breast cancer cell lines or melanoma. And again, just like we observed in multiple myeloma, if you look into the preferential dependencies for AML, or in this particular case, renal cell carcinoma, these are typically not the type of genes that are differentially overexpressed, mutationally activated, or resident in recurrently amplified regions. There are some exceptions to these genes, and you can imagine that lineage-defining transcription factors for the myeloid lineage or for renal cells can be represented among the top overexpressed genes in those tumor types. But the overall point here is that if you didn't have access to the functional genomic data of the CRISPR assessment, it would have been very difficult to predict what are these candidate preferential dependencies for each one of these neoplasias. You cannot just do it by assessment of the omics data without the functional information. In closing, I wanted to mention that this concept of lineage preferential dependencies in heme malignancies, endocrine neoplasias, reproductive organ neoplasias, or other neoplasias does not necessarily mean that a lineage preferential dependency is a top dependency for a given tumor. It may not be exclusively essential for that tumor type. It may be shared by other neoplasias, especially the ones that share a lot of biological similarities, and may not even be the best therapeutic target. But the point of identifying these targets is that if there are inhibitors, if there are perturbagens against them, the identification of these myeloma preferential dependencies or preferential dependencies for other targets indicates that those inhibitors, those therapeutics merit a very strong consideration for testing in those specific diseases for which these targets have a functionally preferential role. And so in summary, I think we were very pleased to be able to use functional genomics technologies to identify so many new promising candidate targets, and at the same time re-identify known targets and mediators of action for all the drugs that have preferential clinical activity in myeloma. This gives us more reassurance that the many understudied and underappreciated targets revealed from this effort may be promising. And because of the fact that these preferential dependencies cannot be readily identified by conventional omics technologies and omics molecular profiling approaches, we feel that they have the potential to have therapeutic relevance both for high and for standard risk multiple myeloma. And in fact, the experience with thalidomide derivatives, proteasome inhibitors, and other myeloma preferential dependency targeting therapeutics is that they can work both in standard and high risk settings. The fact that these dependencies were not identifiable readily by established molecular profiling methods at the genome, transcriptome, or epigenome level is something that is, I think, very important to us, and it highlights why we have to continue using these functional approaches. And specifically for the myeloma preferential dependencies, some of them may have applications in other conditions, but also in autoimmune disorders through the targeting of non-malignant plasma cells. And in closing, for those of you that have an interest in neoplasias of reproductive organs, thyroid cancer, renal cell carcinoma, maybe pancreatic cancer or liposarcoma, and not just heme malignancies, that concept of myeloma preferential dependencies, I think, is very relevant to all of your efforts. And I would look forward to collaborating with you if anyone has an interest in taking some of this knowledge closer to projects near and dear to your hearts. Thank you for your attention. And again, many thanks to all the people that made this work possible and inspired this work. And I look forward to answering any question that you may have for me.
Great. Thank you, Constantine. We can take questions now if you want to put them in the chat or address them. I'll address one to Constantine. So Constantine, it strikes me, if you're going to accept the premise that these cancer cell lines are reflecting biology that is reflective of sort of the innate tumor biology, why not go directly to drug screens and apply a very similar analysis comparing, drugs, instead of looking at drug vulnerabilities, instead of looking at selective gene dependencies?
That's an excellent point. And in fact, in the paper, in the supplement of the supplement, I don't know if Tanya is on the call, but there are some journals where there is limitation to how many supplemental figures you can have. So now you have to have a supplement to the supplement, which we did. And in the supplement of the supplement, there are two panels in which we took data from our collaborators at the Broad from the drug repurposing screens and other screens they have conducted. And we identified that some of the targets that are represented in the screen, I think DOT1L inhibitors came up and some other genes came up. Inhibitors against those targets were identified as having preferential activity against myeloma lines versus non-myeloma lines. So the principle, Tony, that you described is absolutely correct. If we had the chance to test millions and millions of compounds from chemical libraries and do it on all of these systems with, let's say, the PRISM panel, yes, I do believe that we would be in position to re-identify proteasome inhibitors, thalidomide derivatives, etc. There are some caveats. I think one caveat is, unfortunately, many of the preferential dependencies that we have identified, especially the ones that involve transcription factors, are not yet druggable. And the probability of identifying just from general chemical screens selective and direct inhibitors is going to be low. But I think,
let me forget about genes for a second.
Yeah.
I mean, if you're going to really buy into this concept, you don't need the genes. You just screen the drugs, and that should be enough to identify the stuff that'll selectively work in myeloma patients.
Like I said, we agree. I agree with that principle because we applied it. It is in our study, and I think the chemical libraries that were available for these types of screens that were conducted comparatively in one type of disease versus the other, they were enriched for certain types of certain types of drug classes that had been developed in the past. So, I agree with the principle of what you say. How this is going to be affected more practically and how we will apply it on an individual patient, I think that is a little bit more complex, but I agree with you.
I'll also ask, my understanding is that there is a bias in multiple myeloma cell lines toward the study of plasma cell leukemias, and that most of the cell lines arise from plasma cell leukemia rather from than typically like bone-based or other, medullary myeloma. Do you think this has a risk of skewing the results?
I think the only way to be 100% confident that it doesn't is once we get better at doing some of these CRISPR screens with patient-derived samples. And we are, one of the actually a couple of the different patient-derived xenografts that we've been establishing in that scaffold system that I showed, we've been able to expand some of those cells. We've been developing Cas9 positive versions of those cells, and we hope to be able to do those types of screens. Now, the challenge is that with patient-derived samples, with PDXs, the number of genes that you can test is going to be smaller, but I do think that we are going to be hopefully soon in position to validate in patient-derived samples some of these preferential dependencies. And then the challenge there, as you know, Tony, is that a lot of the patient-derived samples that can be expanded as PDXs are exactly the type of samples that come from the current version of late-stage disease, which may not manifest itself as plasma cell leukemia, but may manifest as, multi-drug refractory multiple myeloma cells derived from the bone marrow aspirate that biologically and transcriptionally are again very similar to plasma cell leukemia. Also, most of the therapies that we first developed for multiple myeloma, we take them in late-stage disease, maybe not plasma cell leukemia, but multi-drug refractory. So I think I can easily see how, in principle, there can be therapeutics that would work great in late-stage disease, but not as great in early-stage disease. But I'm also not aware of many such examples that we have in multiple myeloma. So I would say, just like proteasome inhibitors, thalidomide derivatives were preclinical developed or examined first in late-stage disease, cell lines, those types of preclinical models, and then their administration became more prevalent in early stage disease. I do hope that whatever new therapies will come out of this effort against myeloma preferential dependencies, if they work against late stage disease, I do anticipate that they will also work for early stage disease. In principle, though, you're right, plasma cell leukemia and multi-drug refractory multiple myeloma is not the same as newly diagnosed disease.
All right, thanks. The hour is late, but I'll open it up to the field and say, are there any questions for Dr. Mitsiades? With that, I think we can close. Thank you very much, Constantine. And we can close out this, and I will see you all next month. So long.